Is the audio on? I
Good
can't
morning, everyone. This is day 7, very exciting day. You have heard a lot about spillover and all, but today we are going to have very focused talks on outbreaks, spillovers, but also from the One Health perspective. So the first speaker for today, you might have seen Dr. Guha Dharmarajan for the last few days. He's a doctor. He's done his master's and all in veterinary science, but he never practiced it. He's done PhD from Purdue University, and he's been working on vector and malaria mosquitoes really as a model system for a long time. And currently he's a professor in Korea University, teaching the mechanism for spillovers and outbreaks and biostatistics, how you reason science behind it. So over to you. Looking forward to your talk. Thank you.
Okay. So I've never actually worked with guinea pigs, but we have an expert here. So the idea today, so what happened was that I actually made a PowerPoint presentation. I'm not completely lazy, so I did make one. But then, as I kept sitting through every talk, including, thank you, Arinjay, what I noticed was that like every one of my slides had already been kind of covered, right? So then I kept on like deleting slides and adding more slides and everything. At one point, I just gave up, right? I said, I still have another 3 days, right? So so I said, okay, fine. What I'm going to do is that I'm going to wing it, right? And so that's what we are doing today. So the hope is that, again, it's a completely it's an experiment, so we're all scientists here. So the whole point is that, if it fails, we will probably learn a lot more from it than if it succeeds. But I'm hoping, you don't start an experiment hoping that you'll fail and learn. You start an experiment hoping that you'll succeed and get that Nature or Science paper, right? So that's what the hope is for today. Okay. So what I was trying to do is to so we've kind of gone way into the depths, right, of each of these different mechanisms. So what I wanted to do was to kind of take like a kind of a bird's eye view, right, of this kind of process which we call spillover, right? So the minute that we're talking about a spillover, again, I apologize for this. It's a terrible human, but anyway, I'm not able to get the human to look any better. Maybe if I put an eye in the mouth, yeah, okay. But when the minute that we're talking about spillover, it doesn't have to be a human, right? I'm just using a human as kind of like a model, kind of a novel host, right, for a kind of a parasite. We'll come to what we mean by a parasite and all in a bit. But the minute that we're talking about a spillover, you obviously need some other kind of a host, right? And that host, again, right, could be any kind of a host. So since we are just out of the whole SARS-CoV epidemic or pandemic, let's just use SARS as the kind of the example, right? So we know that, okay, fine, SARS-CoV-2 was originally the ancestral host was the bat, and that through some kind of, again, we don't know what this kind of amplification host was, but this is what is called a reservoir host, right? So we don't know what that amplification host was, but somehow it moved from this reservoir host, which is the horseshoe bat, into an amplification host, and then finally into humans, right? Okay. So we do know this, right? So does anybody know what these kind of diseases are called that go from, or pathogens are called that go from animal reservoir to humans? Zoonotic, right, very good, okay. So these are all what are called zoonotic diseases. Are there any diseases that go from humans to animals, or never? Sorry? Zooanthroponosis, yeah. So basically you have some diseases. Can you give an example of a disease that goes from humans to animals? Tuberculosis, great example, especially elephants. In India, temple elephants are especially susceptible to tuberculosis, bad hygiene, high numbers of people coming in close contact, right? So it's a major kind of a disease that's going from humans to elephants. So what we are mainly thinking or going to be looking at today is what is called spillover, right? And basically that means that a pathogen has gone from this kind of a reservoir host, it's adapted in some way to a novel host, and then it has kind of emerged in that novel host, right? There are different types of spillovers, right? So for example, you could have spillovers that have to happen every time. So I'm mainly talking about zoonotic diseases that are going from animals to humans. So you can have these kind of pathogens that have to go from animals every time because human-to-human transmission is usually pretty low, right? So for example, Ebola. Ebola is a great example of this. You usually have these these kind of epidemics that flare up, they die down simply because human-to-human transmission is pretty low, right? SARS-CoV-2 was kind of important because once it got into humans, it adapted so that it was able to transmit between humans very efficiently, right? So that's the thing. But you also have another thing which is called spillback, right? Does anybody know what a spillback is? Just follow the arrows, maybe.
A disease that goes from an animal to human, and again same disease.
Perfect. Exactly, right? So a disease that goes from the animals to a human, then goes back. So in SARS-CoV-2, is there a good example of a spillback that has been demonstrated? Sorry?
Deer.
Was it deer? But deer is a dead-end host. So deers and cats are both dead-end hosts, right? So they can't actually, again, transmit back to humans. So that's a dead-end, to my knowledge. Of course, we keep on getting new information on SARS, so I don't know. But the mink, sorry? Yeah. But the mink was one of those in Europe which was a very efficient spillback host, right? So that's the thing. Okay. So when we are talking about these kind of diseases, right? Can we just talk about disease just from a perspective of this naive host and a reservoir host and an amplification host? What else do you need for a disease? A pathogen, right? Okay, fair enough. So I'm going to do this. How's that? Okay. So you have a parasite. So ecologists tend to call everything a parasite, right? So for example, veterinarians will call a pathogen like a bacteria and virus and everything else, and parasites are only the larger parasites. But ecologists tend to group everything into one term, which is parasite. So can you give me some examples of parasites? Tapeworms, very good, okay. Any other? Amoeba, great. Okay. Anything else? Sorry? Plasmodium is another great one, okay. Anything else? Trypanosome, very good, okay. I'm running out of space. What else? So, again, as I said, ecologists are not differentiating. So, most of you are kind of mainly focusing on these kind of eukaryote or like lower prokaryotes, right? What about bacteria and viruses, right? So, all of these, right? Whether we're talking about rabies, or we're talking about brucella, right? All of these, from an ecological point of view, is considered to be a parasite. But ecologists do differentiate between all of these and things like tapeworms. Mosquitoes are another parasite, right? Because they do take a blood meal. Ticks are another parasite. They do take a blood meal. There can be hyperparasites, right? So there are parasites that parasitize parasites. So for example, Plasmodium, right? The definitive host of Plasmodium, where the sexual reproduction takes place, is actually the mosquito, right? So these are what are called hyperparasites. Okay, fair enough. And so ecologists call these what are called microparasites, and these what are called macroparasites, right? So what is the major difference between these microparasites and macroparasites? Size, very good, okay. Are there any other ecological differences other than size that can differentiate a microparasite and a macroparasite? Sorry? Number per host, very good, right? Usually, microparasites you have millions and millions of microparasites inside a host, while in the case of macroparasites, you could have hundreds or even thousands, but not in the millions, right? Another very important point is that microparasites actually reproduce in the host, right? They have multiple generations within a single host. Macroparasites generally do not. There are, of course, some macroparasites that do, but for the most part, for example, if you look at a tapeworm, the definitive host is a human. The tapeworm is, of course, a hermaphrodite, so it can self-fertilize, but regardless, so it has sexual reproduction. Then those proglottids come out into the feces, and then it's eaten either by a cow in the case of Saginata, Taenia saginata, or the pig in the case of Taenia solium. But then that becomes a hydaticis, and then a human has to eat that uncooked meat to get the parasite, right? But only a single generation of this parasite will be in one human, right? It can have multiple rounds of reproduction, but it's a single generation. Okay? Okay. So now I want to take a step back and ask you, what is a parasite? From a purely ecological point of view, right? So we've already said that I cannot define a parasite. Well, actually, let me hold that thought for just a second so that I can finish this. So is there anything else? So we've talked about these kind of biotic factors that are going to be affecting this kind of spillover, right? We haven't come to any of the mechanisms or anything else. We're just talking big picture right now. Are there any other factors that can affect whether or not a spillover is going to happen? Perfect, right? So environment, very good. So we have environment. Let's just put a pin in that. We'll come back to this in a bit. But now what I wanted to do is, again, ask, what is a parasite? Right? So remember, we can talk about the environment completely on its own. I can talk about weather, I can talk about climate. The Earth would probably have weather and climate with no living organisms, right? Okay. I cannot talk about a host unless I talk about the ecology of a host, unless I talk about the environment, right? Because the host depends upon the environment. The host's entire fitness, evolutionary path depends upon the environment it's in, along with all of the other biotic interactions and everything else, right? I cannot talk about a parasite without talking about the host and the environment, right? So you can see that this is kind of like a hierarchical set of things. So if I cannot talk about a parasite except in light of a host, what is a parasite? How do you define a parasite? Very good, perfect, almost perfect example, right? Or definition. Something that depends upon another organism for resources. Okay, so is that other organism happy to give the resources? Oh, you want resources? Here, I'll give it to you.
Doesn't give its services back to the...
It doesn't give any services back, right? So if it gives services back, what will it be called? Sorry?
Mutualism.
Mutualist, right? Very good. So now we can see that when we talk about these kind of ecological things, we can have a species 1 and a species, right? And we can kind of classify these interactions into 3, right? This is again a very simplistic perspective, right? But all of you have seen this thing. So, oh, I don't think I like this. Okay, okay, that's fine too. Okay, so when you have a positive and positive, as was mentioned, this is mutualism, right? Okay. What about a negative and a negative? What is that? Competition. Very good. Okay. And this one right here is what ecologists will actually call predation, right? So where one organism is getting benefited, which is the parasite or the predator, and one organism is getting, and one organism is getting negatively affected, right? Okay. So why do ecologists group? So ecologists, if you ask an ecologist, there's very little difference between a parasite, a predator, and a herbivore, right? We will say, okay, fine, a cow is the same as a tiger, is the same as a virus. It's all the same. Why? Because that interaction, from an eco-evo point of view, that interaction is the same. One is getting benefit, one is getting harmed, right? That's it. So, but then what is the difference between a true predator, like a tiger, and a parasite? Perfect, right? So E.O. Wilson, I love E.O. Wilson, right? Nobody can put things better than him. So he says that a parasite is nothing but a predator that eats its prey in units less than 1, right? Predators will go out and they'll eat an entire prey, right? Or they'll kill an entire prey. Whether they eat it or not is up to other ecological forces, but they'll go out and kill an entire prey, right? Okay, fair enough. Parasites do not do that. Herbivores also usually do not do that, right? Okay. So the other very important difference between a true predator and a parasite is the generation time. True predators usually have a generation time either longer than or similar to their prey, while parasites have generation times usually much shorter than their prey, right? Than their host. And why is this important? Why is the generation time important when we talk about evolution? Very good, right? So perfect. So Ashwin showed that perfect example of C. elegans where they showed that sexual reproduction is being maintained because one hypothesis of parasites. Parasites reproduce asexually. They're able to mutate very quickly. They're able to adapt very quickly. If hosts and parasites are in this so-called arms race, this Red Queen hypothesis, where you have to keep running to stay in the same place, then what happens is that the host has to have some way of generating more variation. And how do hosts do that? When their generation time, their mutation rates are so low, through sexual reproduction, right? Okay. So, but why is this sexual? I think ask me some other time about the twofold cost of sex. I always get kind of distracted, then I'll go off herring down some other rabbit hole. Let's focus on spillover dynamics for today. Okay, so let's just finish this because we wanted to this thing. So what is this called? Where you have a zero and a positive? Commensalism, okay. And a zero and a negative? Amensilism, very good, wow. Okay, now, and what about this, 0 and 0? Okay. Okay. Let me tell you, yes?
Does pluralism is actually there
this is exactly what I wanted to mention, and thank you for pointing it out. I am that's why I put those on red, in blue. I'm very skeptical of anything that says 0, right? You can never show no effect, right? You can never show no effect. This is why you have a null hypothesis and you always just reject the null. You can get evidence for an effect, but you cannot get evidence for no effect, right? You may be looking at the wrong variable. So if I say that, species A does not affect species B despite the fact that they are sympatric, that is, they are living in the same area, it may be because I haven't looked at the correct variable where species A is affecting species B, right? It could be positive, it could be negative. It could be very indirect, right? We don't know, right? Okay. So that's why I'm a little skeptical about this. But again, put a pin in this because I will come back to commensalism when we talk about anti-parasite defects, right? So it is not something I want to throw away completely. Yeah.
Things, because in commensalism and amensalism and mutualist things, we are just seeing the single species interactions, A species A and species B. So if we come to neutralism, we can't see because we have to think like it is a bulk things. So the entire environment is depending on the particular species and the particular group of species depending on that entire environment. So that bulkiness, we can't observe that things, right? I thought like that.
Yes and no, because when you look at it, none of these are actually happening in nature, right? These are what they say, right? All models are wrong, some models are useful, right? That's it, right? So the fact of the matter is that this is a useful model to think about how species interact. However, when you look at competition, competition can be affected by parasitism. This is what is called apparent competition. So, for example, we know that parasites have a negative effect on a host, but a parasite, despite having a negative effect on a host, if it has a more negative effect on another host, it may actually increase the competitive ability of one host, right? This is kind of like saying that how fast do I have to run to outrun a tiger? If a tiger is chasing you and me, how fast do I have to run? That's it, right? So all of evolution, all of ecology is all relative. You can't say a parasite is negative unless you put it into perspective. A parasite may have a more negative effect on another host and actually be positive for you, despite reducing your fitness, right? Okay, so we've said what is a parasite. Another simple question, so why do parasites cause harm? Why can't they just be nice people or nice organisms and not cause harm, right? There's so many nice organisms, right? Honeybees are all helping these plants and everything else. Gut microbes helping us to digest our food, right? Doing so much work for us for a pittance of a pay, right? Why do parasites cause harm? To balance whose population? So the parasites. Okay. So do parasites knowingly work to regulate a host population?
No, they're just doing their work. They need to survive. Just they want to extract the nutrients. They are going in their own way.
But why? Why would I want to extract nutrients? Why would I go through all the work of extracting nutrients? Well, then I'll just throw it away. What am I doing with all these nutrients, these resources?
Survival things.
Surviving?
Survive. Yeah.
Okay.
To evolve and survive.
And thank you, right? Okay. One of the most, in my mind, poorly worded statements which has become very, very famous is Darwin's survival of the fittest, right? What is my Darwinian fitness? My Darwinian fitness, despite the fact that I've survived for 50 years is 0. I have no offspring, right? I don't plan to have any offspring. So as far as Darwinian fitness is concerned, just surviving doesn't matter. If I survived for a year and produced 100 offspring, I would be better off than surviving 100 years and producing 1 offspring, right? Of course, assuming that all the offspring survive and everything else, and there's a huge amount of literature on that, R and K selection and everything else. And if we have time, we'll come to that also, because that also actually affects spillover dynamics. Yes?
Target survival means it's not only the individual survival level, it's based on reproduction also, like generation that species should survive.
So, okay, I am a pure Darwinian, right? There are some group selectionists out there. I, for one, have never found very good evidence of group selection working in the absence of individual selection, right? Again, this is, again, a very complex area, and I don't want to get completely sidelined. But in general, all the really good evidence of group selection is when group selection is working in the same direction as individual selection. Kin selection is a good example of that, right? So why does kin selection work? Because I am related. So Haldane famously said, was asked, if somebody was drowning, right, would you jump in and save them? Haldane said, I would jump in and save exactly more than 2 of my full brothers, more than 4 of my half brothers. Why? Because all a question of relatedness, right? I share 50% of my genome with my full sibling, and therefore, if I am able to save more than 2, I'm actually at an advantage, even if I die, right? Okay. So in that sense, yeah.
In the context of sibling, we read this concept. But what about like ecologists or wildlife biologists? They are taking their life to save the other species and all these things.
Okay, so I usually stay away from humans completely, right? Humans are like bats. They are unique and they don't follow the trends, right? So, for example, there are going to be very few species that are able to destroy the planet as efficiently as we are, but there are also very few species that are going to be having so many conferences on how to save the planet, right? Most species will be like, yeah, we are the dominant one, we will dominate, even if in the long run, you are going to go extinct, right? So individual fitness usually trumps over group fitness, and we will come to a thing in parasites just now. If you just hold this thought for a second, right? Okay. So, yes.
Just add to his point. So that thing is known as a reciprocal altruism, we call it, because that one species may save other species, irrespective of it think that, oh, that species will save me in future things. So it may save or it may not save. So the altruism is kind of animal behavior, which it leads to that kin selection, what he told about. The relativeness will means the savingness will decrease from generation to generation, from 50 to 0.25, like 1 to 5, like it will go on decreasing.
Okay, so it's a little different because reciprocal altruism doesn't depend on kin selection simply because by definition, if you're a different species, you are, of course, everything is a tree of life and everything is related. So in that sense, you would agree. But from a purely practical point of view, you're not going to like have a kin benefit by saving another species. But you're right. In many cases, what happens is that there is this idea that I will be repaid at some future date. Again, this is not an area that I'm very familiar with. So I would probably just stay away from commenting. I'm sure that there are other people in this conference who are probably more expert at this. But I wanted to, so we've come to this idea that, okay, fine, we have these parasites, they are going to be negatively affecting these hosts. They're going to be using these resources that they get from the host in some way, right? And we've said that, okay, fine, this way is for reproduction, right? Survival and reproduction, okay. But is it enough for a parasite to just reproduce, right? Well, actually not, right? So for example, if you're thinking about virus, a virus can get to millions of copies in your body, right? When does a virus really become fit? If it just keeps on staying in one body, will it be fit?
Transmit from one to
Exactly. So it has to transmit. So that's what I think Farah was talking about, this R0, R0, right? We generally are measuring our parasite's fitness in terms of our basic reproductive number, and that is the number of new infections that one infected host can lead to, right? Of course, it comes up with a lot of other things, so it has to be in a completely susceptible population and everything else. But basically, this is the metric of parasite fitness. Okay, so if I say, okay, fine, you know, ecologists generally say that, you know, the amount of harm that this parasite is causing, or the amount of resources parasite is getting, we usually measure it in this kind of catch-all term, which is called virulence, right? So if I'm going to be plotting, say, transmission, right? So let's call this a probability. As virulence increases, what do you think will happen to transmission? Will it reduce or will it increase? Will transmission decrease? Why? Why would transmission decrease as your virulence? So the parasite is using more and more host resources, is reproducing more and more. Very good. Okay. So these are two very different things, right? So when we're talking about virulence, they say that we have to talk about 3 T's. First is what is called transmission. The second is what is called trade-off, right? And the third we'll come to in a second. Okay. Which reminds me that I cannot go this far because I have a third. Okay. Let's go this far. Okay. Okay. So generally, as virulence increases, your transmission will increase, right? But as was pointed out, if I am going to be plotting survival, my survival will actually reduce, right? And this is why when we are talking about something else, which is like probability of new infection, right? This probability looks like this. So this is approximately equal to your parasite fitness, right? And you can see that generally the rule of thumb is that your,
It appears to me a little counterintuitive. So
What?
if the hosts are dying,
Yeah.
you expect the disease to die off and the transmissions to go down, no?
Exactly.
Isn't it?
Yes, right? If all the hosts die, right? So this is a probability of dying,
Okay.
right? So there's some probability of survival. So if you say that this probability is say 1, and this is say 0.5, and this is say 0, right? We're not saying that all of them are, there's no probability of survival, okay? So remember, a nice example I tried to give is, think about these parasites, like these, if you think about a virus, right? Think about the virus like these, I don't know, like ants on a waterproof but edible like biscuit, right? These ants are reproducing on this biscuit, which is floating in this water, right? The more they reproduce, the more ants there are, and the more they eat the biscuit, right? The biscuit is waterproof, so if the ants don't eat the biscuit, they're going to survive. There's no problem, right? They eat a little bit. So at what point is it really smart to eat that biscuit very fast and produce large number of little baby ants? Sorry? Perfect, right? If there are a whole lot of empty biscuits floating around, and your likelihood of you coming in contact with this empty biscuit is really high, so you're able to jump to an empty biscuit before the biscuit you're on drowns, right? Okay, so that's an important point, right? So this is why when we talk about this, I told you that there are 3 T's, right? The third T is time. And we already saw data that showed that virulence, this is measured very practically as a case fatality rate, right? One kind of simple measure, right? In the case of SARS-CoV, right? In the beginning, it was like, I don't know, 7% or something like that. But it quickly came down to about 2 to 3%, right? And why do you think virulence, what is the advantage of virulence coming down as the epidemic spreads? What is changing as the epidemic is spreading? More people get immune, right? So basically, you have these biscuits which are either full or they're not accepting anymore, like ants, right? So though you're running into, so the number of biscuits is staying the same, right? But somehow these biscuits now have some coating on them that prevents the ants from eating them, right? So you may come in contact with the biscuit, but those biscuits are not habitable for you, right? So what is happening is that you are, again, remember, I speak very generically and very loosely, and this is a very bad idea, right? It is not that the pathogens are deciding to reduce their virulence or anything of that sort, right? I use these terms very loosely and very badly, but what's happening is that pathogens that have lower virulence are able to survive on that host for longer and therefore increase their likelihood of coming in contact with a susceptible host as that proportion of susceptible hosts keeps on reducing, right? Remember, so when we were talking, so in the beginning, wow, I'm way behind, okay, I haven't started, okay. In the beginning, right, when we were talking about SARS-CoV, it was actually a droplet infection, right? So aerosol only came up, I think, at Delta, right? So it was droplet infection. So at that point, right, this whole masking and everything else was actually not to protect you from getting infected, but to prevent you from spreading the infection, right? So therefore, the masking actually, theoretically, would have helped to reduce the virulence of the pathogen simply because since the pathogen is not able to get out of the host, pathogens that were able to stay in the host without killing them were more fit than ones that were killing the host quickly, right? So therefore, there are multiple reasons. Vaccination was another reason, of course, for the reduced virulence, but there are multiple reasons that this thing. But in general, what the data was shown, I don't know who showed that data on this thing, but the virulence had started falling well before vaccination started, right? So it wasn't that when vaccination started, the virulence started falling. The virulence had started falling well before, right? Okay. So we've come out to these two things. So we have this thing, okay? So now the question that I have is whether I'll finish anything at all. Okay. Okay.
Does it apply to every organism or every pathogen?
Sorry?
That virulence things, does it apply to every pathogen?
Very good question. No. Simple answer, right? The beauty of ecology, right, is that I can say no to any answer. any question, right? Because I can always find an example that will be counter, right? So let us take a step back, right? And I'll give you a small story to put this whole thing into perspective. And this idea of whether this survival and transmission are always linked, right? This has been a cornerstone of immunology, it's been a cornerstone of evolutionary biology, it's been a cornerstone of ecology, right? That when we look at a host and a parasite, there's always this kind of antagonism, right? When the parasite benefits, the host has to be harmed. When the host reduces harm, the parasite has less fitness, right? So there's this kind of antagonistic coevolution that is happening, this Red Queen kind of hypothesis. Yeah.
Arm race. Right?
Hi, how are you?
Arm race.
Yeah, sorry? Arms race, exactly. Okay, so the question is that always the case? Okay, let me tell you a small story. So assume, right, that you're a hardworking field ecologist, right? You've gone out to the field, you've done a whole day's of work, right? On your way back, you find that you have 100 rupees in your pocket, right? And you decide to buy one khati roll, right? You say, well, you know, it's not great, but I can only spend 50 rupees. I can buy one khati roll. You go home, you're sitting down at your table, right? And you hear a knock, and you find that your neighbor comes in and eats half your khati roll, right? The neighbor is getting a benefit, you're getting a harm. Would you call this hypothetical neighbor a parasite? It's an apocryphal story, so you shouldn't think too much, right? The answer is, yeah, I mean, from an ecological perspective, you can consider them to be a parasite, right? So what is the origin of the term parasite? Has anybody thought about that? Does para mean different? Parallel is what? Beside, right? Next to, right? Okay. And what is site? Yeah, so this is always a little tricky. You can't completely just translate these from Greek to English that easily. So site actually comes from sitos, which is wheat or food. So basically a parasite is somebody who sits beside you and eats your food, right? So that is the origin of the term parasite. Okay. So now you have this parasite neighbor, right, who you know is going to come and eat your food. So the next day, again, you go out, you're again doing your field work and everything else, and you're again coming back and you say that, you know, you get paid this 100 rupees, so you're going to buy another khati roll, right? And then you suddenly remember that that crazy parasite neighbor is going to eat half my khati roll, right? So you're trying to figure out, what am I going to do, right? So while you're going, you see this unemployed ninja, right, who's standing there, and you say, okay, fine, you know, hey ninja, if I pay you 50 rupees, can I employ you so that you can keep this parasite neighbor out, right? So ninja says, sure, give me the 50 rupees. Ninja comes along and is inside the thing. So when the neighbor knocks, ninja shows sword, neighbor runs away, you eat your full khati roll in peace, happy, right? Okay. Third day, you do the same thing, you say this is working out well, but now the neighbor has learned. So the neighbor evades the ninja, comes in, and is trying to eat your food. When the ninja takes out the sword and starts like slashing the neighbor and everything else, fine, they kill the neighbor, but unfortunately, they also maim you, right? So now you're in a real dilemma, right? You're going back the fourth day now, and you're thinking, okay, fine, so I can buy this, I have 100 rupees, I can buy the khaki roll for 50, I can hire this ninja for 50, but this ninja is like hurting me also, right? I'm like all bruised and like cut and everything else. is there anything else, any other option that I have that I don't have to hire the ninja, but I can still get my full khati roll? You can buy one more roll, right? In ecological parlance, this hiring a ninja is what is called resistance. Buying another khati roll is what is called tolerance, right? Tolerance, in the case of parasites, when you're tolerant, what you're doing is that you're investing in tissue damage or repairing tissue damage, right? How do parasites cause harm? By damaging your tissues or something of that sort. So for example, if you want to become tolerant to anemia, you can either kill the parasites or you can invest in producing more red blood cells, right? So these are the two kind of broad kind of things that we have in terms of anti-parasite defense for parasites. But remember, if we plot and host fitness, right? So if we say that, okay, fine, you have this, in the case of resistance, if you have a high resistance phenotype, right? That high resistance phenotype will have higher parasite fitness simply because you have lower parasite load. So you're killing parasites, therefore you have a higher fitness. So if you have so this is low resistance, high resistance, right? But you can see that this line is the same, right? So the unit effect, so a unit increase in parasite fitness causes a unit decrease in your host fitness, right? The only difference is that because your overall parasite load is lower, right? Your overall parasite fitness is reduced and your overall host fitness is increased when you have high resistance. Yeah? In the case of tolerance, it's a very different, right? Again, you have the two things. You have your parasite fitness, right? So if you have a low tolerance genotype, again, you have something that looks like this. So for a unit increase in your parasite fitness, your host fitness is reducing. However, in the case of tolerance, what happens is if you have high tolerance, just because the parasite load increases or the parasite fitness increases or anything else, your fitness may not change at all, right? So, but the important point is that both resistance and tolerance will increase host fitness in the face of a parasite attack. So they are both anti-parasite defense mechanisms. The difference is evolutionary. In the case of resistance, you set up this red queen dynamic, right? You have an antagonistic coevolution because an increase in parasite fitness automatically reduces host fitness, right? In the case of tolerance, however, you don't set up a red queen dynamic. You could have complete kind of no kind of coevolution between a host and a parasite. And once again, a completely tolerant host, you will not be able to differentiate a parasite from a commensal, right? Because the parasite is getting some benefit, but the host is not facing any harm, right? Yeah. Okay. So that's a big question. So it looks like it's been going both ways. So there are some commensals which have become parasitic. There are some parasites which have become commensals. So I don't think there's any like universal kind of answer.
So my point of question is when Varicella zoster in case of chicken pox. So after curing, after the clearing of that symptoms, it will again in the future it will come in the form of herpes. So what do you call that thing? Is it the Varicella zoster as a dormant? Is it tolerant to that host, or our body is resistant, and again it is appearing again.
Okay, so as I said, I'm very good at this kind of like bird's eye view. My knowledge of very specific things is usually pretty bad. So when you ask me a very specific question about chickenpox and herpes and everything else, I actually don't know the answer, right? Because my answer will be, as a good ecologist, it depends, right? So,
like going deep into that action, but I'm just curiosity of that. What is that pattern we call as? How it is adapting? I'm going in the sense of
becoming herpes?
Yeah, a vessel zooster is going dormant to that particular some stage, and it will again develop as herpes. You know with anything that things?
Yeah. So you're saying that once you have chickenpox, you're more susceptible to become infected with herpes?
The chickenpox will turn to form as herpes.
Really?
In future.
Yeah,
shingles.
Oh,
shingles. Yeah.
Yeah.
The receptor sites are common.
Common, yeah.
Okay, so herpes simplex can be latent, so it can be in your lymph node for a long time in your life. When immunity goes down, it jumps. Okay, varicella zoster doesn't have that kind of work on herpes virus, but both are herpes virus. One is 7 and other simplex is 7, and varicella zoster is different. But it triggers. Actually, it reduces the immunity. When the immunity reduces, both will jump up.
So I'm asking, what is this kind of pattern is called?
It doesn't have a pattern actually. I don't think so, because we are working on herpes virus in elephants. Okay. So we see, because there are different. Sorry. Guha.
No, you're good.
Different strains of them. They can come at any time.
Okay.
So they are not cross-reacting. Antibody can not cross-reacting.
Yeah, because that is my question.
So I'll have to. Because this is actually very different from where we're going. So we have to cover quite a lot of ground still, and I only have half an hour. I'm sorry to this thing, but hold your question, right? So, again, I'm not very good at these very specific details about individual diseases. I'm more of a big picture kind of person. So let's focus on the big picture right now. Okay. So then when we are talking about this, right, we have already looked at this resistance and tolerance. And so basically, we know that if we're going to be plotting virulence, right, generally what theoretical models and kind of empirical data is showing is that when we are looking at resistance, you will find that as virulence increases, your resistance to the disease increases. And the reason for that is, as I was telling you, when the ninja is like fighting and everything else, they tend to hurt you, and that is what is called immune pathology, right? There is some kind of a cost to activating that immune system, and which can cause self-harm, right? So the only time that you're going to be willing, again, evolutionary, that harm is when the parasite itself is highly virulent, right? However, when a parasite is very low virulence, right, you generally tend to get tolerance, right? So as virulence increases, tolerance reduces, resistance increases. So you can see that usually, right, at this kind of intermediate virulence, both resistance and tolerance will be working, right? But this is in a nice co-evolved system. Unfortunately, when you have a spillover, right, you don't have that co-evolutionary history. So then what you can get are these kind of bizarre situations where you actually have very high resistance despite the fact that the parasite has very low virulence, right? For example, SARS-CoV-2, right? SARS-CoV-2 actually has a relatively low kind of a virulence, right? But most of the severe SARS-CoV is because of the cytokine storm. This is a maladaptive kind of a response. So it's this excessive kind of a response leading to inflammation and lung damage and everything else, right? So in many ways, this immune pathology is much more harmful than the parasite itself, right? But that usually only happens when you have you don't have this kind of coevolutionary history, right? So you've all heard about the Red Queen. This is what is called the suicide king, right? So basically, the parasite kind of damages itself with this kind of a situation. And so you have a situation where you have a maladaptive response leading to your own kind of mortality, right? So you're basically committing suicide despite the fact that you actually don't have to, right? Okay. So the other thing that I wanted to mention was that things get a little more complicated, right? Why do they get more complicated? Because if I put my resistance to tolerance, right, is, let's say, resistance greater than tolerance, or tolerance is greater than resistance, right? And then if I just look at like parasite fitness versus host fitness. So how is parasite fitness correlated with host fitness, right? This can either be positive or it can be negative. When resistance is greater than tolerance, you're usually going to get something that looks like this. So you have your parasite fitness is negatively correlated with your host fitness. When tolerance is greater than resistance, you'll get something like this, where it's generally positive. So therefore, there's this break between your transmission risk and your survival. So just because a parasite's fitness increases doesn't necessarily mean that the host fitness reduces. In some cases, a host fitness can even increase, right? For example, in the case of mosquitoes, right? If a mosquito survives the infection without reducing parasite load, it is more likely to transmit the infection. It's more likely to take its blood meal, right? So the mosquito benefits and the parasite benefits, right? So you could have a situation where you actually have a positive effect. Okay, so this is what I wanted to kind of talk about as, okay, so these are all factors related to the parasite. So this we'll call virulence. This I'm just going to call something like persistence, right? And what do I mean by persistence? I mean that if you're infected, if you can retain that infection for a very long time, you are more likely to be a good reservoir host. This is why bats are such good reservoir hosts. So we were seeing yesterday about this persistent infections without much clinical pathology, right? This makes bats a wonderful reservoir host. The other issue is that in ecological systems, you actually have a situation where bats can become like these kind of mixing pots, right? So you have more recombination inside a bat because you have these like multiple generations of the virus and everything else. So you can actually get these kind of novel kind of viruses coming up in a bat because of the longevity of the bat along with the longevity of that infection, right? So you can have these kind of situations coming up. But what about an environmental pathogen? Do environmentally transmitted pathogens, for example, anthrax, do they have to care about this longevity? How do they do it? Environmental pathogen is one that is transmitted through the environment. So basically, organism excretes it, it's in the environment, then a contaminated environment is picked up by another organism. So it's not direct contact, it's not through a vector, it's not through any biotic means, it's through completely abiotic means. So what do these environmental pathogens do? How do they survive, or how do they persist? Dormancy, right? Anthrax spores can stay dormant for decades, right? They're increasingly finding, so we can look at persistence from the thing of, so in environmental pathogens, we can say spores, right? But we can also say these glaciers, they're saying could contain these kind of pathogens which have been preserved for millennia. And as climate change is increasing, these glaciers start melting, it is possible that these old pathogens get released and we are now exposed to pathogens which we are not adapted to at all, right? So there are multiple ways in which these kind of things can lead to spillovers and new kind of infections. Okay, so we've been talking about all of these different things. We've got these viruses, we've got bacteria, we've got protozoa, we've got helminths. So when we are talking about,
So can I say that this resistance is more like when it's an immune response, and tolerance is more like when the host is making more resources for the parasite as well.
So resistance is exactly the immune response. So it's a very classical thing, but we don't really differentiate between humoral, cellular, because all has the same overall effect of reducing the parasite's fitness and increasing host fitness.
Okay.
Yeah, okay. So when we're talking about spillover risk, so basically parasites that have low virulence, that tend to lead to a tolerant reaction, are going to be more likely to spill over because they're going to lead to these persistent kind of infections, right? But when we talk about these various groups of parasites, do all these various groups of parasites have the similar risk of spilling over? So which parasites are most likely to jump to a new host? Sorry? RNA viruses, perfect, right? Very good. So we can say, if we are going to say something like spillover risk, I'll definitely put RNA viruses right on top, right? Then, no, so we just talked about the taxa. We'll come to the route of transmission in a bit. Just the different taxa. So RNA viruses are the most likely to spill over, I agree. What's after that? Bacteria would be way down here somewhere. Okay. What else? What other kinds of pathogens are there? Protozoans, right? They're usually lower than even bacteria. What else? Sorry? Bacteriophages, let's not get into bacteriophages. They're a whole other cup of tea. I'll talk a little bit about them if you give me a second, but let's stick to the pathogens of vertebrates, right? Yeah. Okay. Protozoa, then? Fungi, okay. I actually don't know where fungi fall in this whole thing. Let me do something that I do know, right? Then you have the single-stranded DNA and then the double-stranded DNA. So, but why? Why do we have this particular order of this kind of spillover risk? Why is it that it goes from RNA viruses to single-stranded DNA, double-stranded DNA, bacteria, protozoa, and then helminths? Why is it this particular order? What is driving this particular order in which they spill over, or the risk of spillover? Mutation rate, very good, right? So if you put mutation rate, right, minus 10 all the way up to, I don't know, minus 4, this is 10 raised to, right? So RNA viruses have about a mutation rate of 10 raised to the power minus 4. DNA viruses, I don't know, somewhere here, then here. This is, I think DNA are much lower. So it's like 10 raised to the power minus 8, right? Bacteria are actually much, much lower. They're like 10 raised to the power minus 9, right? And all of these eukaryotes are actually very, very low mutation rate, right? Whether they are prokaryotes, like single cell or multicellular, right? So basically, you do have this situation where mutation rate does affect your spillover risk, right? But there is something else. So why is it that you don't have viruses? Do you have any organism that is mutating at a rate much greater than 10 to the power minus 4? So you have something which are called virions, right? Now these virions are bizarre things. You only get to them when you're studying plants, right? These are a relic of the RNA world. They don't have any capsule. They just have this RNA molecule that's just floating around, and it just gets inserted into it. Prions are a bit more complex because they're actually misfolded proteins. So I don't even know what a prion is. Is it really life? It's kind of like a crystal, right? So if you throw a crystal into a solution which has that thing, you will get the growth of a crystal, right? Is that what it is? Prions are a bit odd because I can't really say, is it mutation, right? What is causing this prion to evolve? I have no idea. Again, I know what I know, and I know what I don't know. So I'll stay away from prions if you don't mind. But virions, right? Where do they fall in this thing of spillover risk? No idea, right? Virions could be anywhere. For the most part, oddly enough, virions are about 200, 300 nucleotides long. They're very small, right? A single base pair change can make them either infective or non-infective, right? You actually find that most virions are quite host-specific, right? There are few virions that infect multiple hosts. They mainly only infect angiosperms. They don't infect others. We haven't found a virion of animals, for example, right? So what is happening here? It is possible that at some point, right, you start seeing a decrease in your spillover risk simply through this thing which is called DFE, which is this distribution of fitness effects. So basically, most mutations are deleterious, and when you have very high mutation rates, you're just not going to be able to survive very long. Your fitness overall reduces. You're not going to be able to do much, right? So it's possible that that's what is happening there. Okay, so finally, I did want to talk about something which was quite important, at least to me, and that is that why is it that, so we are talking about these spillover risk and everything else, and you're saying that, okay, fine, there are some pathogens that are capable of moving from one species to another. Some pathogens just don't move, right? Why? What is there a cost? Why would I, as a pathogen, right, unless there's a cost to not being a generalist, why won't I just go ahead and infect everybody I come in contact, right? What is this cost of being a generalist? Why would you not be a generalist? Very good, right? It comes down to competition, right? Why is it coming down to competition? It's the same reason I went and got a PhD, right? I said, okay, fine, I'm going to get a PhD, I'm going to become an expert in this field, then I will outcompete anybody who is in this small field of disease ecology. That's it, right? I'm the tallest one here in disease ecology. Right? Then if you tell me, oh, hey, I have a question for you on immunology, I'll say, well, I'm not really that good at immunology. Then you say, what about cell biology? Kind of, sort of, right? They say, what about literature? I have no idea. I read books, but I can't answer anything, right? So you can see that specialists, right, they outcompete generalists in their area of specialty, but anywhere outside, they're going to be outcompeted, right? So if you have a situation where you have, this is what is called, you all heard about the niche, the Hutchinsonian idea of this n-dimensional hypervolume and everything else. What is one major factor that determines the niche of a parasite? The host, right? So if you have, say, host 1, host 2, and host 3, right? A specialist parasite will do really well in host 1. So this is parasite 1, or let me call it specialist 1, specialist 2, specialist 3, right? A generalist parasite, on the other hand, will kind of do okay on all the hosts, but in the presence of a specialist, the generalist is going to be outcompeted. So the generalist only does well if the specialist is removed. If all the 3 specialists are removed, the generalists do reasonably well on all the parasites, but while these specialists are there, it can never outcompete them, right? Yes?
I was also thinking of some chance component to it, because to be a generalist, you'll need multiple, like some combination of multiple mutations. So
Very true, right? So this is where it usually is what they think is happening, right? There's a cost to plasticity, right? Each host has a slightly different kind of a defense mechanism or something of that sort. You cannot, if you have to optimize yourself for all 3 hosts, then you have to optimize for 3 different things. If you're trying to get an average, you're never going to be exact, right? So this is what they think is happening, that there's a cost to being too plastic, right? But the fact of the matter is that in the end, what you have is that you have these, so you have a generalist here, which can kind of outcompete only in the absence of these specialists, right? So generalism is useful, but it doesn't necessarily kind of guarantee your success, right? There's one more thing I wanted to mention. Yeah, okay. So we have another 5 minutes, so I wanted to, you can see that I'm still on the parasite. I was supposed to finish host and then the environment, okay? So in the next 5 minutes, I'm going to do everything. Okay, I don't think so, but somebody was mentioning about transmission route, right? And I think that this is a very important point. So let us talk a bit about transmission routes, right? Okay. So what are the different types of transmission routes that we can have for parasites? How are parasites transmitted from one host to another? How was COVID transmitted? Respiratory, right? So you can have a respiratory kind of a thing. 1 second. I want to get a little more space here. Okay. Yeah. Okay. So you have respiratory. Anything else? Sorry? You'll have to speak up, people. Don't worry about mic. Just speak up. Yeah. Sorry? Yeah. Oral. Yeah. So you can go through diet. Yeah. What else? Sorry? Body fluids, right? So just direct contact with some kind of body fluid or infected material, right? Saliva, in the case of rabies. If a dog bites you, you'll get rabies, right? Yeah, sure. Anything else? Fomites, yeah? So fomites also, I would probably just put this in here, right? So fomites are relatively large. They cannot be inhaled, so they're in the environment. You touch, then you either eat it or you thing, but it's through some kind of contact. Yeah, good, okay. Sorry? Vectors, right? Okay. So this could be mosquitoes or ticks, right? Or sandflies or, okay, kissing bugs. Okay, let's not get there. Okay, but basically we have we have oral or environment, right? We have direct, that is you come in contact with some kind of infected material, and then you have something which is called aerosol. Okay, so if I wanted to, again, let us just talk about this spillover risk, right? If I wanted to plot this, in general, you will find that vector-borne diseases tend to be the most number. Actually, let me not say spillover risk, this is a bad thing. Let me just say number zoonotic, right? Because that's where the data comes from. I don't want to mix my terminology. So if you just look at the number of zoonotic pathogens or parasites that we have identified in the past that have successfully moved from animals to humans, we find that most are vector-borne. So 25, depending upon the source you look at, it goes anywhere from 25 to 40%, right, are vector-borne. Oral is little less than that, direct transmission is less than that, and aerosol is even less than that, right? So you have a decreasing thing. So we always think that aerosol is really important. Why? Because we have COVID in our heads, and we think that, oh my God, aerosol. But remember, we're talking about wildlife, right? You have to come within kissing distance of a bat to get your COVID. This is why those wildlife markets were so important for SARS-CoV. COVID-19, it hasn't been, confirmed. No one will confirm or deny. I believe that wildlife markets did have some role to play, but at least in SARS-CoV-2, the original SARS, wildlife markets were the thing. In the case of MERS, it was these camel farms, which were right next to humans, that were the kind of amplification location. So basically, in these kind of situations, you have high human density and high density of these amplification hosts. In the case of SARS, it was these civets, which are being maintained in these wildlife markets, right? And so you have this situation where you can have this kind of transmission. Yeah.
The more than direct.
Sorry?
Aerosol should be the more than direct.
So we are only, again, that's what I was saying. So we're talking about the number of zoonotic pathogens that have been detected. So I'll give Ansil a series of papers that all of this data is from. So this is not something I'm just making up. There's some readings that I can give to you, and if you have any questions, you can definitely ask, right? Okay. So we have all of this, right? We have do we have another minute or so, Ansil? Yeah? Okay. Okay. Now I wanted to talk briefly about these so-called reservoir hosts, right? And what are the factors that could contribute to making a really good reservoir, right? So what do you think some of these factors could be? Okay, why don't we just think of, when you think of a reservoir host, what are some of the taxa that you think about? Orders of, let's just stick to mammals, simple. Orders of mammals that are important reservoirs of zoonotic infections. Chiroptera, Rodentia, primates, okay, fair enough. So you have bats, rodents, primates, and anything else? Carnivora, right? These pretty much make up the 4 major orders where you have high zoonotic potential. Okay. So if I was to put a graph and I asked you just like that, number of zoonotic parasites, what is the order you think these will be in? Would bats be number 1? Very good, right? So generally, they find that rodents have the most number of these things. Bats and primates are very close, but bats have a little more. Then you have carnivores. And then you have the ungulates. Ungulates generally are very low, right? Okay. Let's just stick to mammals. Birds are, again, but generally because of the diversity of birds. So diversity of birds hold that thought. Let me stick to mammals because birds, again, is too diverse a group to just be put into one kind of a bucket. These are all mammals, so it becomes a little easier to make these kind of comparisons. Okay, so then why is this is one very simple explanation that people have come up with is that it's just the total number of parasites that these orders have, right? So generally, you'll find that it's almost a linear relationship. So bats and primates, again, are very close to each other. Carnivores are here, ungulates are here, right? So you have a situation where there's almost a linear relationship between the number of parasites that are there in the order and the total number of zoonotic parasites that are there in that particular order, right? So, for example, rodents have somewhere between like 100, 120 zoonotic or total parasites. They have about like 40. Again, these are all, incomplete data sets. So, but it's about like 40 to 50% of the total parasites are zoonotic in the case of rodents, right? Okay, so that's one reason. But the problem, so what is the problem of just saying, hey, all rodents have so many parasites, right? Can we really compare all of these orders? Why should we not compare these different orders? What do they differ in? What do you mean closeness? Okay, phylogenetic closeness, right? So this is a very important point. What happens in these kind of data sets is that you are conflating the diversity inside an order with the parasite diversity. So diversity begets diversity. The more diversity you have in the host, the more diversity you'll get in parasites, right? So, for example, rodents have what? Like greater than 2,000? Bats have greater than 1,000? Primates are about what, 500? Carnivores, 300? Ungulates, if you combine horses and equines and bovids and everything, will probably be about 600. Okay? So you can see that obviously rodents will have a large number of parasites, and therefore they also have a large number of zoonotic parasites simply because they have over 2,000 species. So you cannot really compare, right? So you can see that if you start looking at it on a per species perspective, so for example, if I put something like number of zoonotic hosts divided by total diversity, right? Total number of species. So now we're talking about like how many, what is the proportion, what is the probability that if I just picked one of these species from these orders, that I will have a host that has a zoonotic pathogen, right? What you will find is what taxon will be right up on top, you think, per species? Sorry? So if I'm just looking at the number of zoonotic hosts per species, not for the whole order, which order will have the highest number of zoonotic pathogens per species? Will it be rodents? So you can see that rodents have about 2,000, so that's about right. Bats have about 1,000. Primates, right? So when we start looking at it per species, or the probability that a particular thing is then you will find that primates have primacy, right? Why are there so many zoonotic primate hosts? Somebody already mentioned phylogenetic proximity, right? Primates, if we put phylogenetic distance, right? Primates are, I don't know, close to 0, something is 1 somewhere here, right? And you can see that if you are going to do this kind of a graph, what you will find is that you have primates, rodents are the next most this thing, and they have a high level of parasites, so they'll come somewhere here. Your carnivores and bats come somewhere close to each other. And of course, your ungulates are the loser every time, right? So, but you'll have something that looks like this, where you have the most numbers per of potential things here, and your ungulates are last. Very good. Okay. Why don't we take a break? And I'm not sure if anybody needs a board, but what I'll probably do, since I'm like only halfway through, but we had some nice discussions, is that I will probably just skip my research talk and just continue on this. And then I can just upload the slides for the research talk, and then if you have any questions, we can do this thing. I'll probably just get to the kind of core message that I wanted to talk to you about for the research talk right at the end. But if you can just keep that like not, or this thing, then we can just continue in the, I think it's what, 4:30 to? Yeah, 4. Yeah. Thank you so much, everyone. So it was fun, and hopefully we'll continue in the evening. Yeah.